Then I'll introduce today's speaker, Bastien. I think most of you already know him. He's been a member of the IB community and fairly well integrated into the MVC as well for a number of years now, based on his experience in the Huelsenbeck Lab. Just to give you a little bit of his background, he earned his PhD in Lyon, where he was working on the early evolution of growth temperatures. Okay. And then afterwards, he got a position at the National Center for, well, it's the CNRS, which stands for the National Center for Research in Science in France, which is a peer research position there. But he postponed that position in order to come and take a postdoc with John Huelsenbeck, which provided many of us an opportunity to get to know him. Now he's back to give this seminar, and he's going to be telling us about his work developing methods for whole genome sequencing phylogenetic analyses. Nice. Welcome back, Bastien.
Thank you. So this talk is going to be a little bit, hopefully not messy, but eclectic, and I will discuss various topics, and hopefully you will get something interesting out of one of those topics, at least. And so basically I'm interested in genome evolution, and if I try to summarize what factors I'm interested in that influence genomes, I come up with a list of basically 4 factors. So the phylogeny of organisms, the molecular processes that include metabolism, but also that include the mechanisms of replication or transcription, these types of mechanisms. Then you have the environment that exerts a pressure on the organism, and then you can see effects of this pressure on the genomes. And then you have the demographics, such as the population size in a species, the effective population size, the way the species mate, etc. And so all those factors exert an influence on genomes, and they leave a trace in genomes through different evolutionary processes. And so among those evolutionary processes, you've got insertions of deletions of small bits of DNA. You also have substitutions. You also have duplications or losses of parts of the genome. And finally, you can also have transfers of a part of a genome from one species to another species. And so all those processes leave traces of those factors through evolution. And what I'm interested in is to try to look at extant genomes and try to infer things about those different factors. And so to do this inference, I need to develop probabilistic models of those evolutionary processes. And to do the inference, I use statistics and big computers. So in this talk, I will be just focusing on 3 types of evolutionary processes: substitutions, duplications and losses of genes, and transfers of genes. So I will not be dealing with insertions or deletions. So here's the plan of this talk. As I said, I'm going to be talking about different projects. First, I will be talking about projects that deal mainly with events of substitution. And then I will switch gears to talk about events of gene duplications and losses. And finally, I will add to gene duplications and losses gene transfers. So first, as I said, let's focus on substitutions. And in terms of substitutions, I'm going to be focusing on a particular type of substitution, which include non-synonymous substitutions, so substitutions that change the amino acid of the protein, and synonymous substitutions that do not change the amino acid of the protein. And if you get to obtain those non-synonymous substitutions and synonymous substitutions, you can compute the ratio, DN/DS. And this DN/DS ratio tells us something about the strength of selection. And that's why I'm going to be interested in this DN/DS ratio in the next few slides. So basically, the non-synonymous mutations are more likely to be deleterious than the mutations that do not change the amino acid. And so because of that, you expect that when selection is strong and efficient in a population, you should see lower DN/DS than when selection is weak, because you should see fewer substitutions that are deleterious. So what I'm going to do is that I'm going to take a phylogeny and try to compute the DN/DS ratio on different branches of this phylogeny and see if that can tell us something about the organism that we have in this phylogeny. Yeah. So by mapping these events of substitution, of non-synonymous and synonymous substitutions, we're going to see when selection was strong and when selection was weak on the tree. So usually, when people do that, they use a software that is well known and whose name is PAML. And the way you do an analysis using PAML is that first you need to assemble a data set, so that means an alignment and a species tree, or a tree at least. And then you need to define two types of branches. So in the PAML nomenclature, they are known as foreground and background branches. So there you need to have an hypothesis in mind where you think that there are two types of branches. Branches where evolution is going to occur according to a certain DNDS, where you assume that selection has been of a certain strength. And so you're going to call those branches foreground branches. And then another type of branches where you assume that selection has been of a different nature, less strong or stronger. And those branches are going to be background. And then once you have those two types of branches, you're going to compare the DNDS on the foreground branches versus the background branches. And so this approach has been used widely. But it has, in my opinion, two drawbacks, which is that you can't use it in all situations. So for instance, you can't use it when you don't know a priori what branches should be foreground and what branches should be background. When you have no hypothesis about what's happening exactly in your phylogeny and you just want to explore your data, you can't really use this type of analysis. And then the second problem with this analysis is that maybe you have more than two categories of branch. You just don't have strong and weak selection, but maybe you have strong, mild, and really weak selection. And so in this case, you would want more than just foreground and background categories. So to try to circumvent these problems, we developed with colleagues in Montpellier, in Marburg, in Germany, we developed pieces of software which are called MapNH and TestNH. And so MapNH allows you to just map events of substitution along a phylogenetic tree. And so those events of substitutions can be DNDS, which is what we are going to be interested in the next few slides, but you can also map substitutions that are towards GC, if you're interested in the evolution of GC content along the tree, towards AT. You can also look at amino acid sequences and also map those substitutions between amino acids if you're interested in that. And it's a model-based approach, and it's fast. It's also accurate if we compare its inference to PAML, the software that is basically the reference. So there on the y-axis you've got our software MapNH, and you've got PAML on the x-axis. And basically you see that there's a nice correlation. You can also see that there's quite a spread in the PAML inferences of the DNDS ratio. So to me that's probably not the bad news for our software, because you see that some of you may see that the values at the bottom there are very high. So for instance, you can have a DNDS of 100 there on this line. And so what this is telling us is that PAML is inferring a lot of cases where the DNDS ratio is about 100. Whereas MapNH does not infer cases where the DNDS ratio is about 100. And so this is to me kind of a good news, because the DNDS ratio of 100 is really extreme, and it looks more to me like an optimization problem than a real biological inference. So overall, MapNH seems to be pretty robust and agrees pretty well with the reference software PAML. So we have this piece of software, and then we have another piece of software that uses the output of MapNH to just find clusters of branches along which sequences have evolved similarly. So basically, with these two pieces of software, you don't need to define a priori background and foreground branches, but the software is going to come up with background, foreground, and maybe middle ground branches if it finds that there are three types of branches in your phylogeny. So basically, we've been using this software on particular phylogeny. So what you need to do is map those non-synonymous and synonymous substitutions on a tree, recover branches along which evolution has been similar, and then you need to interpret the findings in the light of characteristics of the species that you have in your phylogeny. So if you have questions at any moment, you feel free to interrupt me. So in this case, the species we've been interested in are frogs, and in this case, mentally frogs from Madagascar. So more precisely, we chose 17 species of frogs. We extracted 4 gene sequences. we aligned them and concatenated them, built a tree using FiML, and then we applied our approach to cluster branches according to the DNDS ratio on those branches, on the phylogenetic.
Bastien, can I ask a question?
Yeah.
Can you estimate the tree using one set of markers and then infer your DNDS ratios for another set that's not included in the phylogeny estimation?
Yeah. You give it a tree, you give it an alignment, and if the tree and the alignment do not come from the same data, it's going to be okay.
No problem.
As long as the names match. And so here are the results we obtained when we applied this clustering approach of branches according to their DNDS. And so you can see that basically we have two colors on this tree. The black is just not a color, it's just that we have no data for this branch. So we have two colors, we have red and we have blue. So this means that we have two clusters. We have one cluster of red branches and one cluster of blue branches. The blue branches happen to have a DNDS of 0.04 and the red branches have a DNDS of 0.08. So the red branches have a DNDS that is higher than the blue branches, meaning that selection on the red branches has been weaker than on the blue branches. And so when you look at these three, you find that, so first of all we have those two clusters, but those two clusters seem to be somewhat not randomly scattered along the tree. We kind of see a cluster of red branches and clusters of blue branches. And so to try to understand what may be associated to those clusters, I circled in red species that happen to breed in streams. And so those species, circled in red, happen to breed in streams. And the other species that have not been circled happen to breed in ponds. So I know basically nothing about frogs, but reading the literature about these frogs, it seems to be that the stream breeders have a more fragmented habitat. In fact, they notably tend to speciate more often than the pond breeders. And the explanation I suggest is this following one. Basically, if you're a stream breeder, you have a fragmented habitat. And if you have a fragmented habitat, you may have a lower effective population size because basically you have barriers to gene flow. And because you have a lower effective population size, then selection is less efficient. And if selection is less efficient, then that means that the DNDS is going to be higher. So that's what you would be seeing here. Basically, stream breeders where habitat is fragmented, selection is less efficient, and DNDS is higher, and you have those red branches there. So that's the hypothesis I can come up with to try to explain this pattern. Obviously, more tests are needed to confirm or reject this hypothesis.
And what happens if there's incomplete sampling in the group? Like, how does that affect things?
I'm sure there's incomplete sampling there. I make no assumption about a model of speciation on the tree. It's just the topology and then mapping substitutions on that. So we don't have the type of problems that we have when we try to infer radiation parameters such as extinction and speciation rates. So I think it should be fairly robust to that.
And how does your model deal with recombination?
It doesn't. It ignores it. So when I concatenate all the genes together, I'm making the strong assumption that the evolution of those genes all those genes have followed the same tree.
So another model that comes to mind is, and I wonder how this fits into everything, is that you could have a selections change, right? You could have a dramatic change in the environment or something that would drive your DNDS ratio up by positive selection, as opposed to by weak selection.
Yeah.
And so is that alternative hypothesis something that can be addressed in any way? There will be cases where you have a strong shift in the environment that will cause organisms to adapt. And in this case, you may be able to find signs of positive selection. There, we are analyzing 4 different genes, so that would be a strong, if it's positive selection, maybe it's an effect that has affected several sites differently among those 4 genes. So to me, it seems a little bit less easy to explain this pattern with positive selection than with just relaxation of selection, because you need to come up with an argument that says there has been a change and then this causes positive selection.
Right, maybe not so likely for frogs, but maybe if you look like something like viruses.
Oh yeah, yeah.
Yeah,
Yeah.
might be interesting.
Yeah, in viruses you see lots of instances of positive selection. For instance, when you put some medicine in the body. So basically, with this approach, we have first agnostically clustered the branches, and then this led us to propose some hypotheses about the ecology of those species. And then this hypothesis, we can test it with more experiments. But obviously, we can also test different types of hypotheses, and the next example I want to tackle is going to just deal with genome evolution. So basically, we are going to be trying to correlate events of substitutions with events of duplications and losses. So as I said, events of substitutions and events of duplications and losses. And we're going to try to see whether there's a link between substitutions and duplications and losses. In fact, I'm going to test a particular hypothesis using mitochondrial genomes of geckos and of frogs. So the same frogs as before, but I'm going to add in genomes of geckos as well. So the hypothesis I want to test has been proposed by Michael Lynch in 2003 and deals with the idea that genomes increases in size through drift and not natural selection. So basically, when you have, in his model, when you have gene duplications that increase the size of a genome, it's not because they are initially beneficial to the organism, but instead initially they are deleterious, and if they fix in a species, it's because it's through drift and not through natural selection. So at the time this hypothesis was proposed, this was a pretty strong change to what people thought about gene duplications, which were thought to be adaptive mostly. And so basically what we wanted to do in this paper with Matt Fujita and Jeremy Brown, who were also at DMBZ, was to test this model with mitochondrial genomes that have undergone duplications. So those mitochondrial genomes were from, again, the frogs that I showed you before, as well as geckos. And so the geckos were Heteronotia binoi geckos, which happen to have two types of individuals, the sexual individuals and the asexual individuals. And interestingly, we find the duplications only in the mitochondrial genomes of asexual geckos. It's somewhat of a mild mystery why we find only duplications in the asexual geckos and not in the sexual geckos. And so what we've been doing there is just to compare the DNDS on branches with duplications versus branches without duplications. So there we don't need to cluster the branches agnostically because we already have the hypothesis because we know we basically can infer fairly confidently where duplications have occurred on the phylogeny and where no duplication has occurred on the phylogeny. And so we can cluster the branches in those two clusters, branches with duplications and branches without duplications. So we did this test using 3 different data sets. So 2 data sets for the geckos, one with a lot of individuals, but only 1 gene, and another with fewer individuals, but more genes. So 13 individuals and 10 genes for the second data set. And then we used the same data set of mentelid frog as before. And so we compared two models. One model where we assume that all branches have the same DNDS, so same selection pattern over all branches. And one model where we assume that the selection has been different on branches where you have duplications versus branches where you do not have the duplications. And so what we find when we do a statistical test is that we favor each time the model where we have 2 clusters of branches. So there seems to be a difference in selective pressure between branches with duplications and branches without duplications. More precisely, the DNDS on branches with duplications is larger than on branches without duplications. So that means that duplications tend to fix when deleterious mutations, non-synonymous substitutions, fix. That's telling us that duplications fix in episodes of weaker selection. And so basically this supports the idea of Michael Lynch that duplications can fix more through drift than through selection. Because when selection is weaker, then you have more drift, and you so happen to see that you have more duplications. So basically, these results support the idea that duplications can arise in genomes through drift. And basically, the idea that genome complexity arises through drift is supported by these results. So to conclude on this part where we focused on substitutions, so I presented first a software to cluster branches of a phylogenetic tree based on those substitutions. And then I hope I showed some examples where we can use those substitutions to provide insight into ancient selection, the patterns of selection among branches of a phylogeny. But you can also use this type of approach to study the evolution of growth temperature. As Jim told, I worked on that a few years ago. It was among bacteria and archaea, so not the type of organisms you're mostly interested in. And you can also, and there are lots of examples in the literature. I just cherry-picked examples that I was closest to, where you study various aspects of the biology of the organisms based on substitutions. And so if you do not know this paper by Nicolas Artio and Raphaël Pujol, I suggest you take a look at that. It's really interesting. So now, as I said when I described the plan, we're going to switch gears and focus on duplications and losses. So it's another type of evolutionary process that those factors leave through, they leave traces through duplications and losses as well. And in this case, I'm going to talk about a project where we are trying to co-estimate gene trees and species trees. And I'm going to focus on mammalian phylogeny as an example of this approach. So basically, when one wants to study genome evolution, there are two types of objects that are really useful. First, the species tree, which I will represent like that, and then thousands of gene trees. In a given genome, you've got thousands of genes, and because of that, you get thousands of gene trees. And if you want to understand the entire dynamic of genome evolution, it's really useful to have gene trees for all those gene trees. And so what we're going to try to do in this part of the talk is try to come up with a method that can reconstruct the species tree and the gene trees to give us some insight into genome evolution. And so this has to meet two challenges. So first, we have to do large-scale computations because, as I said, we have thousands of gene trees, and these days we have dozens of species, if not hundreds of species. And then the more fundamental problem is that we have a strong interdependence between the species tree and the gene tree. Basically, if you know the species tree, it helps for reconstructing the gene tree. And if you know gene trees, it helps for reconstructing the species tree. And usually, when people want to reconstruct, for instance, a good species tree, they just cherry-pick a few genes and just go this way from the gene trees to the species tree. And then the other way around, when you're just interested in the evolution of a particular gene family, because you know that the gene has a function of interest, then the best approach is just rely on a species tree that is assumed to be known, and just go this way. Using the species tree that is known, you try to infer the best gene tree that you can come up with. So both these approaches tend to ignore the two-way dependency that exists between the species tree and the gene tree. So we wanted to come up with a method that would be able to jointly reconstruct the gene trees and the species tree according to this interdependency. Now I'm going to explain what I mean by this interdependency more precisely. So there we've got the species tree with 4 species A to D. So we have 4 genomes in those 4 species, and in each of those genomes we have genes. And so for instance, some gene family will have a very simple evolution. So for instance, this gene family here has a phylogenetic tree that very closely resembles the species tree. Okay? But then gene trees can have a more complex evolution. For instance, they may have undergone events of duplication, such as this one there. And so because there has been an event of duplication there, we have 2 genes in species A and 2 genes in species B. We can further complicate that by adding a gene loss in an ancestor of species B, or by instead having an event of lateral gene transfer from an ancestor of species AB to an ancestor of species C. And finally, we also have incomplete lineage sorting that causes gene trees to differ from the species tree. So overall, we have different processes that make the gene trees different from the species tree. But if you're interested in understanding genome dynamics and how they evolve, you need to understand, and you need to be able to have accurate gene trees and accurate species tree to try to come up with how many duplications occurred on a given branch of the species tree, for instance. And so in this part of the talk, I'm going to be focusing on these processes, the processes of duplications and the processes of losses. So all those gene families I can deal with. The gene families that have undergone lateral gene transfers or incomplete lineage sorting, I'm likely to do some mistakes with those gene families. However, in the third part, I'm going to tell you about a method that deals with those families in addition to those three. And for ILS, I'm working on that, so it's not ready yet. But hopefully we'll have that soon. So basically the software we came up with just needs as input gene family alignments. So that can be thousands of gene family alignments. And based on those gene family alignments only, it's going to jointly reconstruct the species tree, there, the gene trees, and the parameters of duplications and losses on each branch of the species tree. And so the hope with this software is that you can learn something about genome dynamics with, for instance, having the numbers of duplications and losses on each branch of the species tree. And this is done in a statistical framework where you have a probabilistic model of evolution for those genes given a species tree. So we implemented this software and this method and decided to use it on, we made some simulations to ensure that it was working properly, and decided to use it on mammalian genomes because we have 36 mammalian genomes in the Ensemble 57 database. We have more now, but at the time we did this analysis there were 36. We selected about 7,000 gene families, so a large number of gene families that were selected just based on their not having more than, I think, 150 sequences or something like that. And so when we ran Phylog on this dataset, we obtained this species tree. So I will not enter in the details of this particular species tree, but to me this species tree looks in fairly good agreement with the current consensus about the mammalian phylogeny. So it's properly rooted because the Phylog can root a tree without an outgroup, just by using events of duplications and losses. And then you recover all the major groups of mammals that have been described. And then inside each group, sometimes you have some oddities, but most of the relationships among those mammals are pretty much in agreement with what we believe of the current phylogeny of mammals. So Phylog basically finds a pretty good species tree. And then we went on to assess the quality of the gene trees. So contrary to simulations where we know the gene trees that we want to reconstruct, so it's easy to see whether we're doing a good job or not, in this case we do not know the true gene trees. So we have to come up with ways to measure the quality of gene trees without knowing the true gene trees. We're going to be measuring the quality of gene trees for 3 different methods, our method, as well as 2 competing methods, one of which is used to build the Ensemble comparable database. So if you go on internet and look at this database and look at the trees they come from this method. And so we're going to be coming with 2 measures of the quality of gene trees. One which is going to be looking at ancestral genome sizes, and the other one which is going to be based on how well one can recover ancestral synthenies, basically how well one can reconstruct ancestral chromosomes using those reconstructed gene trees. So it's a method that has been published in 2012, and that I will not detail, but it's just a way to measure the quality of the gene trees. So according to the first measure, we know that if we make a mistake in a gene tree, when we reconstruct ancestral genomes using those gene trees, we should obtain larger ancestral genomes. So as soon as you make a mistake in a gene tree, you should have more genes in an ancestral genome. And so basically, the idea behind this measure is that better algorithms should yield smaller ancestral genomes. So we can compare our 3 methods according to the size of the ancestral genomes. So here I've plotted the same tree I represented before, but this time I've added on top of that some information about the size of the ancestral genomes that we reconstruct. So in blue you've got Phylogenetic, our method, in green TreeBest, the method of ensemble, and in red PhyML. And as you can see, the size of ancestral genomes is smaller for Phylogenetic in blue. And in fact, you see another, than for the two other methods. And in fact, you can see maybe another trend, which is that the deeper you go in the phylogeny, the larger the genomes are, especially for PhyML and TreeBest. There, we've got fairly recent nodes, we've got all small genomes. They are very ancient nodes, we've got larger genomes. So this is odd. And to me it tells that there's a problem with those, the gene trees that have been reconstructed using those methods. Unless you believe that the ancestral mammalian genome was much larger than extant genomes, which are about this size in our data set. So to me this is indication that the trees obtained by Phylogenetic are more realistic than the trees obtained using either TreeBest or PhyML. And you can have some aggregate measure of that. If you look at all the genomes and you look at the distribution of ancestral genome sizes using PhyML, TreeBest or Phylogenetic, you see that the genomes obtained by PhyML or TreeBest are much larger than the genomes obtained by Phylogenetic. However, the genomes obtained by Phylogenetic still have a size that is larger than the size obtained for extant genomes. So it's likely that there is still some error in our gene trees, unless ancestral genomes are really larger than the genomes that we observe today. So the second measure we used was to reconstruct ancestral chromosomes and use this idea that if you make a mistake in the gene tree reconstruction, using this method, you're going to break synteny groups, which means that you're going to break basically chromosomes. And so what we should have is that better algorithms should yield more genes in ancestral chromosomes, ancestral synteny groups. So when we look at that, we obtain this graph. Again, Phylogenetic is in blue. This time, the larger the better. So again, according to this metric, Phylogenetic does better than TreeBest and PhyML. It finds more genes in ancestral synteny groups than isolated in their own chromosome. So this metric as well tends to suggest that Phylogenetic trees are better than TreeBest trees or PhyMLs. And in fact, if you look at a particular example of a tree that is correct according to this measure and not correct according to this measure in TreeBest, you obtain the following results. So on the right, you've got a gene tree reconstructed by Phylogenetic, and on the left, you've got the same gene family, but a tree reconstructed with TreeBest. So this would be the tree that you would download from internet. And as you can see, so I've color-coded the species there. If you look at the colors, you see big blocks on the right, so for the Phylogenetic tree, but you do not see those big blocks on the left. In fact, you see some rodents, then you see dog and the cow, then you see primates, then rodents again, then the horse and the cow, and then again primates, etc. Whereas for the Phylogenetic tree, all the groups of species are monophyletic. So this is kind of explaining why the measures that we have, the genome obesity measure or the ancestral synteny measure, tend to find that Phylogenetic trees are better than the TreeBest trees. And I would say that according to my own sense of what a good tree is, this tree seems to be better than this tree, which is really scattered and for which you need to assume a very complex history of duplications and losses to explain this weird pattern. Okay, so to conclude on this part, we've developed a software, Phylogenetic, that you can use with whole genome data. It's meant to be used for thousands of genes. And it provides a good rooted species tree without need for an outgroup. It provides rooted gene trees for thousands of gene families. So if you're interested in gene family evolution or even the evolution of a particular gene family, this software could be useful to you. And it also allows you to get accurate numbers of duplications and losses, although I've not shown you the results for this part there. And because of that, you may be able to learn something about species evolution, genome evolution, and gene family evolution when you use Phylogenetic on your data. And it can be used on large data sets. So this was the second part where I was dealing with duplications and losses, and now I'm going to
end this talk by adding to the mix gene transfers. So now we are going to come up with a model that deals with duplications, losses, and transfers. So being in the Museum of Vertebrate Zoology, you may have little interest in gene transfers, but I'm going to try to convince you that it's still interesting. As I said, it's the third part where we use gene transfers. And I think gene transfers are interesting, in part because you can think of gene transfers, of the problem of gene transfers and species phylogenics, as very similar to the problem of parasites and hosts. So if you're not interested in gene transfers, you may still be interested in parasites and hosts. And if you try to draw the parallel between this problem of gene and species with horizontal transfers and this other problem of parasites and hosts, then you can come up with the idea that gene transfers is basically like host shift. You've got a gene coming from one species to another species. It's much like a parasite coming from one host to another host. Then you can see gene duplication as parasite speciation. Let's say you are inside a given genome. If you have a duplication, then you have two genes inside this genome. But if you're inside a parasite inside a host and you speciate, then maybe you have two species of parasites inside the same host. And then you have also this last equivalency between gene loss and parasite extinction. When a gene is lost, it's much like when a parasite is lost inside a host. So I think the models that we come up with for modeling duplications, transfers, and losses of genes can be used as well for modeling host shifts, parasite speciation, and parasite extinction in a parasite-host framework. And then the other reason why gene transfers are interesting, I think, is because they are quite widespread in the tree of life. So obviously they are frequent in bacteria and archaea. They are also frequent in unicellular eukaryotes. They are frequent in deloid rotifers, which do not have sexual reproduction and kind of use transfers as instead of sex, basically. And then you also have transfers that have been documented between, for instance, fungi and aphid. Transfers among plant mitochondria that are pretty common. There have been also documented cases of transfers of transposable elements between different Drosophila species. And recently, there have been reports of a transfer of an antifreeze protein between fishes. And those fishes were not were something like more than 100 million years apart from each other, so it's quite an amazing transfer there. And then there was also a report of a transfer from a trypanosome to human. The transferred DNA sequence from the trypanosome to human was then inherited by the children of the person who got the transfer. So overall, it seems like transfers are pretty ubiquitous, and it's useful for our model of genome evolution to include them to try to get a complete picture of genome. And then another interesting thing about transfers is that they allow us to date claims. So to try to illustrate this point, let's look at this species tree. So we have the same species tree on the left and on the right, but we want to know which of those two nodes is the oldest one. So in this case, we have the red node that is younger than the green node. In that case, it's the reverse. The red node is older than the green node. Now, if we have a model of gene transfers, we can identify gene transfers. And for instance, if we have a transfer from a descendant of the green node to an ancestor of the red node, then this transfer is consistent with this topology, but it is not consistent with this topology because it requires that we go backwards in time. So short of a time machine, we can't do that, and we need to say that this scenario is not compatible with this transfer. So basically, if we are able to model transfers and identify transfers, then we can start to order nodes relatively to each other, and that should help us for dating species trees. So more formally, if we look at a given gene family, such as this one, which has genes in species A, B, two genes in species C and D, one copy there, one copy there, one copy there, one copy there,
And if we have two different competing species trees, so this one where this node is older than that node, and this one where it's the reverse, this node is younger than that node, then to try to explain this particular gene phylogeny given this species tree, we need to assume that there was one transfer event there, under this species tree, whereas if we have this configuration with this node younger than this one, we need to assume that there has been at least two transfers. So thanks to this idea, we can discriminate between different time orders on a given species tree. And in fact, we can also route the trees because different routes will give us different numbers of events. So what we've done is that we've implemented a model that's kind of described there, where we account for events of duplications, transfers, and losses. And we have also events of speciations. And so we've implemented all these things in a program, and now with this program we can try to infer species tree given collections of gene trees or given collections of alignments. The work I'm going to present now is just based on fixed gene trees and reconstructs maximum likelihood species tree in which nodes have been time ordered. And so, as a demonstration, we decided to use it on cyanobacteria, which are interesting in bacteria because they have the best fossil record available, which is not much, but still something compared to the other bacteria. So we use 36 complete genomes and about 8,000 families of homologous genes, which represent a large number of genes in total. And so when we use this program on this data, we obtain this species tree, which again is very much in agreement with the current ideas of the classification of cyanobacteria. It's very much in agreement except for one thing, which is the root of this species tree. As I said, with transfers we can root the tree, and when we do that, we find a root that is not in agreement with what usual techniques based on an outgroup species tend to find. But we think that this root is not absurd, because when we look at the trees under this root, they tend to look much more clock-like than when you use the outgroup technique. And when you use the outgroup technique, if you look at the phylogeny and the branch length, it looks like it could be a case consistent with long branch attraction artifact, where you have long branches at the root. So overall, this new routing is not totally absurd. And then we also looked at the different node ages in this phylogeny, and we compared that to the fossil record. And again, the node ages on this phylogeny are consistent with the few fossils that we have in the fossil record. Now, as I said, we have some information on the orders of nodes. So for instance, this program tells us that node 3 there is older than node 16. And it's telling us that because it found lots of transfers from descendants of node 3 to before node 16, to above node 16. Okay? And in fact, we found several hundreds, as it can be shown there. So if we look again at node 3 and node 16, what we have in this graph is numbers of genes that have been transferred from below node 3 to above node 16. That corresponds to this big blob there. So this big circle is about between 200 and 400, which means that we have about 300 genes that tell us that node 3 should be older than node 16. And finally, with this technique, we can also reconstruct ancestral genomes, just like with the duplication loss model. And when we do that and we map genome sizes along this tree of cyanobacteria, we obtain this graph. So in blue, we've got small genomes, and in red, we've got big genomes. And what we see is that the ancestor of all cyanobacteria seem to have a fairly small genome, but there was a sudden increase from this ancestor of cyanobacteria to the ancestor of all those green cyanobacteria there. In fact, an increase where the genome size was about multiplied by 2. Between 1 and 3, we went from 1,500 to about almost 3,000 genes. And so one thing that we would like to do now is, with this type of approach, where we reconstruct accurate ancestral genomes, we want to look at
what genes were present in those ancestors to try to come up with an idea of what those particular cyanobacteria, in this case, were able to do, what kind of metabolism they had, for instance. The perspectives on this work include a Bayesian method to reconstruct a species tree, on which I'm currently working. We have a method now for integrating over gene tree distributions, and so we do not have to assume that we know the gene trees, whereas in the former work we had to. And we are currently using this method on 102 genomes to reconstruct the tree of life and learn something about the evolution of metabolism, ecology, and environment through the last 4 billion years of evolution. And if you're interested in this type of work, we have postdoctoral positions available. So I'd like to thank you for your attention, as well as my collaborators for their help with these various works. So collaborators from Lyon, collaborators from Europe in general, and collaborators from the NBZ and from the Huelsenbeck Lab. Thanks.